Dear students, let us we study today about the structure of hemoglobin and chlorophyll. In this, we have in earlier class, we have studied about the importance of the metal ions in biological systems. Now, to know about the structure of hemoglobin and chlorophyll, we must know the basic structure of metalloporphyrins. Now let us know what are these metalloporphyrins. Porphyrins are one of the most important groups of bioinorganic compounds in which a metal ion is surrounded by the four nitrogens of the porphyrin ring. Now, to know about this is what the porphyrin ring. So, the porphyrin ring we come across here. So, in this porphyrin ring, suppose in all these cases, they are filled with hydrogen atoms. When it is filled with hydrogen atom, it is called porphyrin. If these hydrogens are replaced by alkyl groups, vinyl groups and some of the other groups, then this porphyrin is called porphyrin. So this is the basic point which you have to remember. So that's why I have said the porphyrin ring or porphine ring contains four nitrogens of porphyrin ring. These porphyrins are made up of four pyrrole rings. So this is A. I have written A is a pyrrole ring, B is a pyrrole ring, C is a pyrrole ring and D is a pyrrole ring. So it contains the four pyrrole rings. Among these four pyrrole rings, here hence these pyrrole rings are bridges through a methane group. So this is what the methane group. So these two are linked, the pyrrole rings are linked through methane groups, CH2 groups. These act as tetradentate ligands. Now when these pyrroles are fused with methane groups, we get like this, and it contains four nitrogen atoms. Hence, it acts as a tetradentate ligand with four nitrogen donor sites. Out of these four nitrogen, two of these are tertiary nitrogen donor positions, which can form coordinate bonds by donating a lone pair of electrons each to the metal ion. So these are the tertiary nitrogen atoms. These nitrogen atoms, they simply donate a lone pair of electron to the central metal ion. And other two nitrogens, these are called the secondary nitrogens. These are known as secondary nitrogens. These they donate the lone pair of electron to the central metal ion and giving out a proton. So thus a porphyrin ring acts as a tetradentate dinegative ligand. Dipositive cation acts as which magnesium ion, ferrous ion or nickel ions are capable of forming neutral complexes with porphyrin. Four pyrrole rings of porphyrin carrying substituents other than hydrogen are called porphyrins, as I have earlier said. The complexes in which a metal ion is held in the porphyrin ring system are called metalloporphyrins. Such complexes play a vital role in biological systems. The size of the central hole of porphyrin ring is perfectly suitable for accommodating metal ions of first transition series or lighter alkaline earth metal ions, giving square planar geometry, and the axial sites are available for other ligands. In this case, I have said, so this is a porphyrin ring. This is a porphyrin ring, and in this porphyrin ring, the iron is a central metal ion. So this iron is a central metal ion, and this forms a square planar complexes. It forms a square planar complexes. Now, the two important metalloporphyrins are heme and chlorophyll. So both heme and chlorophyll have the porphyrin ring in them. Coming to the first one, the structure of the heme. In the structure of the heme, so we have four pyrrole rings. So these are the pyrrole rings. So this is A is a pyrrole ring, B is a pyrrole, C and D are the pyrrole rings. Now in the A ring, we have a methyl and vinyl group. Even in the B pyrrole ring, we have methyl and vinyl group. In the C pyrrole ring, we have a methyl and propionic acid group. And in the D pyrrole ring also we have a methyl and propionic acid group. And at the center, iron is in the form of the +2 state or the ferrous state. Now coming to the iron porphyrins or hemoglobin and myoglobin. There are two iron porphyrins. One is hemoglobin, another is myoglobin. All organisms require oxygen in order to survive. Different proteins have different tendencies to bind and transfer oxygen. These proteins are known as oxygen carriers. The most important two oxygen carrier proteins are hemoglobin, abbreviated as Hb, and myoglobin, abbreviated as Mb. These are iron porphyrin complexes which are oxygen transfer and oxygen storage agents in the blood and muscle tissues respectively. It means hemoglobin carries the oxygen from one part to another and myoglobin stores the oxygen in the muscle tissues. Hemoglobin is a large intracellular protein responsible for the red color to the blood. Myoglobin is a small intracellular protein present in the vertebrate muscles. Hemoglobin picks up oxygen in the lungs and delivers it to the rest of the body. Myoglobin accepts oxygen from the hemoglobin in the muscles and stores it until needed for the energetic purposes. Deoxygenated hemoglobin uses some of its amino groups to fix up carbon dioxide and then transport carbon dioxide back to the lungs. Now here I have shown the structure of myoglobin. So the myoglobin, it basically contains the same porphyrin ring. It is a heme, what I have shown. In this, the myoglobin, the square planar complex of the porphyrin, we have the 4 pyrrole rings. In the first pyrrole ring, abbreviated as A, contains methyl and vinyl group. Second pyrrole ring, that is B, contains once again the methyl and vinyl group. The third pyrrole ring, abbreviated as C, contains methyl and propionic acid group. That is called propionate. It's a COO minus. H is removed. And the D pyrrole ring also contains the methyl and propionate group. And here it is a square planar complex. The 2 more sites are there. In the upper part of the site, oxygen is binded to the ferrous ion, and lower part is binded to the imidazole ring, which is further the link to the histidine moiety. This is what I have written here. Myoglobin consists of one polypeptide chain, that is globin, with one heme group. The peptide chain consists of 150 to 160 amino acid residues folded about the single heme group. Heme is a porphyrin derivative containing 4 pyrrole groups joined by methane bridges. The ferrous ion present at the center of the heme is bounded by 4 porphyrin nitrogen atoms and 1 nitrogen atom from imidazole side chain of histidine residue. This plays an important role in biological fixation of oxygen. Now coming to the structure of hemoglobin. Hemoglobin is a large protein with a molecular weight of about 60,000. It consists of 4 subunits, each of which contains one heme group associated with protein globin. Therefore, in hemoglobin, there are 4 heme groups bounded to 4 protein chains. One heme group with its protein chain is called subunit. The 4 subunits are similar but not identical. 2 subunits form alpha chains of 141 amino acids and 2 form beta chains of 146 amino acids. So, therefore, we have alpha hemoglobin and beta hemoglobin. Both hemoglobin and myoglobin have 5 coordinated ferrous ion. It is bonded by 4 nitrogen atoms from pyrrole rings and 5th nitrogen from imidazole side chain of histidine residue of the protein chain. The 6th position is occupied by water. This is called deoxymyoglobin in myoglobin and deoxyhemoglobin in hemoglobin. However, when the 6th position is occupied by oxygen, then these molecules are called oxyhemoglobin and oxymyoglobin. Coming to the role of hemoglobin and myoglobin. Hemoglobin and myoglobin play very important role in transporting oxygen from lungs to the tissues and carrying carbon dioxide as a bicarbonate ion from tissues to the lungs. Oxygen is inhaled into the lungs at very high pressure, when it binds hemoglobin in the blood, forming oxyhemoglobin, that is HbO2. The oxygen is then transported to tissues where the partial pressure of oxygen is low. The oxygen then gets dissociated from the hemoglobin and diffused to the tissues where myoglobin picks up and stores it until it is needed. Myoglobin has greater affinity for oxygen than hemoglobin. This increases the rate of diffusion of oxygen from the capillaries to the tissues by increasing its solubility. The hemoglobin and carbon dioxide as bicarbonate ion are returned to the lungs from where carbon dioxide is exhaled. Now coming to the structure of chlorophyll. Chlorophyll is a magnesium complex of porphyrin, which plays an important role in photosynthesis. Photosynthesis means production of the food, that is glucose, from carbon dioxide and water in presence of sunlight. During photosynthesis, that is food, glucose and oxygen are produced from carbon dioxide and water in presence of a pigment, chlorophyll and sunlight. In chlorophyll, in addition to substituents, double bond in one of the pyrrole ring is reduced to form a magnesium dihydroporphyrin complex. A cyclopentanone ring is also fixed to a pyrrole ring. Photosynthesis is an important redox reaction occurring in nature to convert water and carbon dioxide into carbohydrate and oxygen in presence of light. This is the structure of chlorophyll. In the structure of chlorophyll, once again we have the four pyrrole rings. Once again we have the four pyrrole rings known as A, B, C, D. This is, and in this each pyrrole ring is linked with methyl and vinyl group, that is the A pyrrole ring. In the B pyrrole ring, instead of methyl group in hemoglobin, we have aldehydic group, and instead of vinyl group in hemoglobin, we have ethyl group. In C pyrrole ring, we have methyl group and we don't have propionic acid group like in hemoglobin. And even in the D pyrrole, we have methyl, and these two, the C pyrrole is linked with the methane and forms a E ring. This is known as cyclopentanone ring. In this cyclopentanone ring is fixed in chlorophyll. This is the difference between the hemoglobin and chlorophyll. And also the D pyrrole, the group, and the C pyrrole group, here we have the two hydrogen atoms. So hence it is called the dihydrido. And the D pyrrole is linked to the CH2, CH2, COOC20, H39 group. So, and at the center, instead of ferrous ion, it contains magnesium ion. So, in chlorophyll, we have 5 cyclic rings, A, B, C, D and E. E is the cyclopentanone group. Now coming to the structure of the heme. So whatever the structure I have drawn there, the same structure is drawn here also. How best it can be modified. So in this structure, these are the pyrrole units. So these are the pyrrole units, and these are the tertiary nitrogens, and this is a secondary nitrogens. So this is how they are linked with each other and forms a heme. And in this, these are the structures of hemoglobin. Hemoglobin, the heme is linked to the so many and forms a globin, a protein, and forms a hemoglobin chain. Now in a structure of the heme, so we have shown here above the hemoglobin molecule and the structure of the heme. Heme is a complex of protoporphyrin and ferrous iron. The iron is held in the center of the heme molecule by bonds of the 4 nitrogen of the protoporphyrin ring. The heme, the ferrous ion, can form 2 additional bonds, one on each side of the porphyrin ring. One of these positions is coordinated to the side chain of histidine residue of the globin molecule, whereas the other position is available to bind oxygen. This is what I have explained earlier. So here I have shown the structure of the hemoglobin. So whatever the structure there, so you go through this, the structure, and the students, you must able to draw the structure of this hemoglobin. So here this is in this, so this is the imidazole ring. This is the imidazole ring. This whole thing is a histidine moiety. So these are the pyrrole rings. This is a methane groups, and oxygen is binded to the air, and you have the methyl and vinyl groups. This is a propionate group. Now coming to the structure of myoglobin and hemoglobin. This is what I have already explained. The same thing in the form of a points is mentioned here. Myoglobin is a monomeric protein that facilitates the diffusion of oxygen in the vertebrates. Hemoglobin is a tetrameric protein that carries oxygen in the blood. Heme consists of tetrapyrrole ring system called protoporphyrin complexed with iron. Heme of myoglobin and hemoglobin binds oxygen for transport. Here I have shown the structure of the myoglobin. Myoglobin is a monomeric protein that is mainly found in muscle tissues. It includes a prosthetic group, the heme group. It can be present in 2 forms, oxymyoglobin and deoxymyoglobin. Oxymyoglobin is oxygen bound, deoxymyoglobin is oxygen free. The tertiary structure of myoglobin contains 8 alpha helicals designated A through H that are connected by short non-helical regions. So here also I have shown about the myoglobin protein structure, and students, you must able to draw this, the myoglobin structure. So here also the informations are given which I have already explained. So these are the iron-rich foods. So the iron-rich foods are tomatoes, pumpkin seeds, so quinoa, green peas, and so on. Many things they have given here. Students, go through these things. Now coming to the how this hemoglobin it function. So the hemoglobin transports oxygen from lungs to tissues. Oxygen diffusion alone is too poor for transport in larger animals. Solubility of oxygen is low in plasma, that is 10 raised to minus 4 molar concentration. But bound to hemoglobin, its concentration becomes just 0.01 molar or that of air. Two alternative oxygen transports, the transporters are hemocyanin, a copper containing protein, and hemoerythrin, a non-heme containing protein. Hemoglobin imparts red color to the blood. It helps to carry out the oxygen and other gases, assisting the respiratory system. It buffers the blood pH and maintains it to the tolerable limits. Sources of physiological active catabolites. The hemoglobin genetic resistance to malaria. Coming to the chlorophyll. Chlorophyll is a vital for photosynthesis, which allows plants to absorb energy from light. Chlorophyll molecules are arranged in and around photosystems that are embedded in the thylakoid membranes of chloroplasts. This pair affects the final functions of chlorophylls, charge separation leading to biosynthesis. Here I have shown the students about the structure of the chlorophyll. In the structure of the chlorophyll, I have shown earlier. So here this contains the, instead of iron, we have a magnesium. And we have A, this is the B, this is the C, this is the D, what you can say, the pyrrole rings. Now, in the A pyrrole ring, we have the methyl group and vinyl group. In the B pyrrole ring, we have the aldehyde group instead of a methyl, and we have the ethyl group. In the C pyrrole group, we have the methyl group, and the D also we have methyl. And in the D, so it is a dihydrogen. Here we have a dihydrogen. So the double bond is broken and two hydrogen atoms are added. And then we have here the E. This is another, what you can say, the ring. So this is called pentanone ring. To this pentanone ring, we have the methyl, that is a COOCH3, the acetate group. The ester group is there. And in the other side, we have hydrocarbon tail. So this is the basic structure of chlorophyll. Now coming to the next structure of chlorophyll molecule. So here we have shown once again the another structure of the chlorophyll. Coming to the properties of the chlorophyll. The chlorophyll is one of the best natural antioxidants. It can be found in many vegetables and fruits, especially those with dark green color. It can help to diminish the high cholesterol content. So, the green vegetables are rich in chlorophyll. So, these are the various vegetables that shows the content of the chlorophylls. which contains basically a magnesium. So the chlorophyll increases the blood count. It soothes inflammation and pain. It improves milk function in lactating mothers. It improves bowel functions and painful hemorrhoids. It improves respiratory function. It reduces and eliminates body odors. It kills bacteria in wounds and spread up healing. It relieves gastric ulcers. So it cleans and detoxifies the body of the carbon dioxide, toxins and stress. It increases the flow of oxygen in the body. It creates an anaerobic environment where the disease cannot live. It alleviates imbalance in blood sugar. So these are all the functions of chlorophyll. Lastly, I have compared the structure of hemoglobin and the structure of chlorophyll. Students, go through this properly. So in this, both have the pyrrole rings. Here we have the pyrrole rings. It also contains the pyrrole ring. In hemoglobin, ferrous ion is there at the center. Here it contains the magnesium ion at the center. So we have the A, B, C, D, the pyrrole rings, and which I have explained in the earlier also. So those things you try to remember in the hemoglobin structure and chlorophyll structure. Keep these two structures side by side and try to understand the similarities and the differences. Students, go through these aspects and try to learn. Thank you.